There are two theories for color vision: pigment theory and opponent theory. Coming to pigment theory, this theory is also known as Young-Helmholtz theory, as it was proposed in 1801 by Thomas Young and the later modified by Hermann von Helmholtz, and it is also known as retinal or trichromatic theory. There are three types of cones, and each has a pigment with different absorbance spectrum. The red sensitive pigment absorbs light maximally in the yellow portion of the spectrum. The green sensitive pigment absorbs maximally in the green portion, and the blue sensitive pigment absorbs maximally in the blue violet portion. Traditionally, the cone pigments are called red, green, and blue respectively. Now they are called long wavelength, middle wavelength, and short wavelength as L, M, and S, and they show optimal sensitivity to lights of wavelengths 560, 530, and 420 nanometers respectively. Each type of cone pigment can absorb a wide range of wavelengths. Also, a particular color light of fixed wavelength produces different levels of stimulation in various cones. The visual cortex compares the relative frequency of action potential in the activated cone pathways and deciphers the wavelength and thereby makes out color. For example, green monochromatic light with a wavelength of 530 nanometer produces excitation of red, green, and blue cones at the ratio of 31:61:36, and it is interpreted as green. The change in the intensity perceived by appreciating the changing wavelength. Stimulation of one type of cone does not encode any color perception. At least two types of cones are required for color vision. According to the Young-Helmholtz theory, the color vision is due to the presence of three types of cone, each maximally sensitive to one of the three primary colors. Therefore, normal color vision depends on the integrity of the cones. Absence or malfunction of one or more than one type of cone cells lead to different forms of color blindness. The fovea mediates color vision as it contains densely packed cones. However, fovea contains only M and L types of cones. Opponent theory. This theory is also known as Hering's theory of color vision, as it was proposed by Hering. It is based on his observation that there are no greenish red or bluish yellow color because they are opposite colors. The red and green colors oppose each other, and the blue and yellow color are opposed. The ganglion cells, the cells of lateral geniculate body, and the neurons of the visual cortex show color opponent property. Single opponent property. The ganglion cells and the cells of LGB are single opponent type. There are two subtypes. Some cells are excited when red stimulus is present in the center of the receptive field and inhibited when green stimulus is present in the periphery of the receptive field. And the other type of cell shows increased discharge rate to green center, and discharge rate decreases in response to a red periphery. They also activated by green periphery and inhibited by red center. So here you can see in the ganglion cell, so when the center is stimulated and it appears as red, and when the periphery is stimulated by the green, it appears red. And in off center, when the periphery is stimulated by red, it appears as red, and in the off center, when the center is stimulated by green, it appears as red. And similarly, it is for the green color. Double opponent property. The cells of the visual cortex are double opponent type. They are of two types. One type of cell is excited by a red center with green periphery, and inhibited by green center with red periphery. The other type of cells excited by green center with red periphery and inhibited by red center with green periphery. So this is what occurs when in on center the center is stimulated by red and the green is stimulated, the periphery is stimulated by green, it is going to appear as red. And similarly, when the center is inhibited by green and the periphery is inhibited by red, it appears red. And similarly for the green in the off center.